So let's just review where we are so far in the Summer Institute. We started with some motivating examples from sociology and psychology about why genetic data is exciting and can matter in social science. David talked about estimating behavior genetics, estimating heritability from twin studies and adoption studies and family designs. That kind of study can be done even without any knowledge without having measured genetic data, and has been done for over 100 years, continues to be done actively today. Then Andrea taught us about molecular genetics and the measurement of genetic data, and I'm going to pick up where Andrea left off and talk about now using that kind of data for gene discovery, which refers to linking specific genotypes with phenotypes. So, I think that's important to understand, not necessarily because you're going to go out and do gene discovery yourself, but because it's the important background to the kind of applications for which you would be using genetic data in the social sciences, and also because many of the issues that come up in gene discovery are exactly the same issues and problems and challenges that arise in applications. So first I want to say something briefly about the kind of genetic data and data sets that we have access to, and this really picks up directly from where Andrea left off talking about the technology. I'm going to say something about how we use that technology, and it gets into the data that we will be analyzing. What you do if you are collecting your own data is you have to get some kind of tissue sample from which you're going to get the DNA. Traditionally, that was blood, and that was difficult and expensive, required a specialist to extract the blood, but you needed blood in order to get high enough quality, a sufficient amount of DNA. Today, what's much more common is saliva, which we now know how to create enough high-quality material from the saliva that we can do reliable genotyping. And so the typical setup is that you have a tube that you give to a participant. They spit into the tube until it gets up to a certain amount of saliva in the sample, and then you can mail it to a lab, and they can do the fancy things that Andrea was talking about. And what we get back from that is a data set, which is a big vector for each individual, say 2.5, if it's genome-wide data. These days, typically, it would be around 2.5 million SNPs, and each entry in that vector for each person is a 0, 1, 2 variable. Andrea also mentioned sequencing, which is not so common today, but is becoming more common as it gets cheaper. And within a few years from now, that is likely to be the more common kind of data, where we will get much larger numbers of variants measured. And that's the reason why it's becoming more common is this is, again, repeating something Andrea showed before, because the costs have been falling very rapidly, although it's still the case that SNP arrays, the genotyping line here in blue, is much less expensive than sequencing technology. So these days you can get the genotyping done for around $30 a person, so to the cost of genotyping that saliva sample. Traditionally, as of a few years ago, the only kind of data sets, or the main kind of data sets that had genome-wide data were either medical genetic data sets that we call cohorts, and that's the term that's used for those kinds of data sets, or twin registries. And typically, the sample sizes are between 500 and 5,000 people. So the medical cohorts were typically formed to study particular diseases like cardiovascular disease or cancer. If you want to access the data, you typically need to collaborate with the people who set up the data set. Some examples are the Framingham Heart Study or the Swedish Twin Registry. So when GWAS started, the earliest GWAS studies that we'll talk about in my next lecture, the earliest studies were all done with data from these sources. And so in order to get enough statistical power to do them, you would need to aggregate information from many of these samples. In the last few years, there have been some large-scale social science surveys that have genotyped their participants. So these are order of magnitude 5,000 to 20,000 people. These are, in many cases, ongoing social science surveys that have been going on. They're panel data sets that have been going on for many years. They've collected a lot of health and social science information, and now they've added genetic data. Most of these are publicly available, subject to, of course, IRB and privacy restrictions. And often the genetic data is downloaded separately from dbGaP, which is an NIH website. So some examples of these data sets are the Health and Retirement Study and the AdHealth data that you're using. A third kind of data set that is also becoming available within the last few years are national biobanks. So the biggest is the UK Biobank with 500,000 people. The Estonian Biobank now has about 50,000 people, but it's growing. And there's initiatives in many countries to create national biobanks, including in the US. The All of Us initiative is essentially a US biobank. These data are also typically available to qualified researchers who go through an application process. The fourth kind of data is also only available to researchers as of a few years ago, and that's data from personal genomics companies. So these are the largest, right? I mean, in the slide I've written 50,000 to 500,000 for both of these types of data, but the personal genomics companies are growing very quickly in their customer bases, so they're going to soon surpass the national biobanks in terms of the number of people that you'll be able to get genetic analysis from. So these are companies where people participate because they want to learn about their ancestry, they want to learn about their disease risks, and then the customers consent to being research participants. And if you as a researcher want to access the data, you have to collaborate with the company, and typically, although some companies have application procedures where, although even so, you may have to collaborate with the company. So it's really specific to the company that we're talking about. So examples are 23andMe and Decode Genetics. Okay. So that's the kind of data that we're going to have in mind for doing gene discovery. So why, so what's exciting about gene discovery relative to, the behavior genetic studies that have been around forever? Well, the heritability estimates that we get out of those studies don't tell us about which genetic variants matter, how much they matter, or why they matter, and they don't allow us to construct variables that we can use in other kinds of analyses. We have to infer the latent variable of the genetic factor or the additive genetic factor, infer properties of it, like how much variance it explains in the population, but we can't use it as a concrete thing in analysis, like controlling for genetics if you want to study something else or interacting it. Okay, so gene discovery is a useful step for doing further research. The causal model that we're going to build on for gene discovery is the additive genetic model that we talked about on the very first day. Just to remind you, there's this additive genetic factor. We have a set of controls so that we can consider the genotypes randomly assigned conditional in the controls. And then there's some noise. And it's going to be helpful often to write it in this sum form because we're interested in the specific beta J's, these average causal effects. That's what we're going to be estimating. Now, this is simplifying this. So the focus on the additive model is abstracting from dominance effects. That you could easily extend this model to include the dominance effects by just adding essentially a second beta for each variant J. That's typically not done in empirical estimation, traditionally because people were just trying to keep the number of parameters small. Although as the data sets get larger, I think that's going to become more common. Another thing that this additive model is abstracting from is the interaction effects, the epistasis. And the reason for that, the reason why when we do the empirical work we're going to abstract from the epistasis is because, as we'll talk about, the multiple hypothesis testing problem is already quite severe, even with the additive model, and the epistasis would dramatically, exponentially expand the number of parameters that we have to estimate. Now, you should worry that when you estimate an additive model, if there are interaction effects, we're going to miss potentially some variants that actually matter if those interaction effects average out to zero. Because what we're getting here is the average effect. The view that the literature has taken commonly is that we're better off, because of the multiple hypothesis testing challenges, we really have to stick with the additive model at first. And if we're interested in interactions, we can focus on them later once we identify the variants that matter. We will miss some this way, but that's a cost that we're willing to accept. So the three challenges that come up in gene discovery are, first, multiple hypothesis testing. So the problem here is that there are typically many variants that we've measured, and in some cases there's even several phenotypes we could study, and there's many possible regression specifications we could estimate or control variables we could include, and so that leads to lots of hypothesis tests that we're running and potentially significant associations that are just by chance. The second problem that's going to come up is population stratification. Patrick's going to go into more detail on population stratification in a later lecture, but the basic idea, which is has come up before, is that there may be gene frequency differences between subgroups in the population. Those allele frequency differences could be correlated with the phenotype for non-genetic reasons, like cultural differences. And so in order to eliminate that problem, we put in our controls, Z, which in theory perfectly deals with the problem because now we have random assignment, but in practice, we often don't have the family fixed effects or the parental genotypes that we would need for that kind of perfect control. And then the question comes up, is the set of controls that we actually use to try to deal with it sufficient? Like, did we actually eliminate the population stratification confound? The third issue is going to be low power, and that's the question of whether our sample size is large enough given the true effect sizes, these beta J's, of linking the genotypes to the phenotypes. And also given our significance threshold and our prior probability, as we'll talk about. Okay, so there are two main approaches to gene discovery that have been pursued in the social sciences. The first is the traditional candidate gene study, which I'm going to focus on in this lecture. And then the second is GWAS, and that's going to have its entire own lecture tomorrow morning. There's a few reasons why I think it's useful to talk about the candidate gene studies. One is for some historical perspective, because the problems that came up in candidate gene studies help to motivate why many of us do GWAS today. A second is that is not actually historical. Actually, most of the work being done in social science genetics today still is candidate gene studies, I think, at least if you count, publications. There's a lot of it going on. And the third thing is that it's going to teach us a lot about things that we need to worry about. Okay, so what is a traditional candidate gene study? It's a study where we focus on a small set of SNPs. We specify ex ante that there's one SNP or 10 SNPs that we're going to study. And they may not be SNPs, by the way. They could also be other kinds of genetic variants. And we focus on those based on some hypothesis that we come up with, that we come into the study with, ideally. And so then these are our candidates for potential association. And then we estimate a simple univariate regression model of our phenotype on the genotype at that genetic variant with our set of controls. We set a significance threshold of typically the standard threshold in the social sciences of 0.05, sometimes adjusted for the number of SNPs that we test. And this, up until 2013, this had been essentially all of the work in social science genetics. And as I said, even today, it's still, I think, the majority of the work. Okay, so this should sound quite sensible to you. I mean, this is, after all, the way that you think of doing science, right? You come up with a hypothesis, and then you test it. And this was the way that most gene discovery work was done, not only in the social sciences, but also in medical genetics, from the earliest days of being able to measure genotypes. So let me say something about the history of what happened in medical genetics, and then I'll say something about the history in the social sciences. So early on, when you could only measure one or a small number of genetic variants at a time, candidate gene studies were all that you could do. And so the very earliest work was candidate gene studies, and there were a number of successes in medical genetics. For example, the association between alcoholism and the alcohol dehydrogenase gene was a candidate gene study. Once linkage studies became possible, and this is something that Andrea was talking about yesterday, linkage increased our ability to find good candidates, because you could do a linkage study in a family with a disease like Alzheimer's disease. You could follow the pedigree and identify some region of the genome where you know that the effect is. But the linkage study identifies a very, very large region of the genome, some big area of a chromosome. In order to make further progress, you then would follow it up with candidate gene studies to look at particular ideas for where the causal variant might be. So a good example of that was the association between Alzheimer's disease and ApoE. ApoE is still the single biggest genetic predictor, the genetic variant, the genetic variant with the single biggest predictive power for Alzheimer's disease. And the way it was discovered was that linkage studies had found a particular region of the genome, and then a medical researcher realized that in that region there was a sequence of DNA that was known to code for beta amyloid proteins, and beta amyloid plaques build up in the brain in patients with Alzheimer's disease. And so that led him to hypothesize that there may be an association, and he tested it, and it indeed turned out to be the case. Now, the heyday of these studies in medical genetics were the 1990s. Toward the end of that period, there was starting to be concern that some of the candidate gene studies, when there were efforts to replicate them, the replications were not consistent. That was not the case for some of these, like BRCA is, in fact, a breast cancer-associated gene. ApoE is associated with Alzheimer's disease. But some of the later ones started to run into trouble. I suspect the reason for that is that these earlier studies were picking low-hanging fruit. They had strong hypotheses, like the ApoE Alzheimer's Association it's a really good biological hypothesis. Like it's coding for a particular thing that's involved in the disease, and then later on, the hypotheses were more tenuous. That's my suspicion about what was happening. And then around 2006, 2007, it became possible to do SNP arrays for the first time, and medical geneticists adopted them pretty quickly. And for some, there was a landmark paper by the Wellcome Trust Case Control Consortium published in 2007 that did the first large-scale GWAS of Crohn's disease, diabetes, and a few other disorders. GWAS quickly became more common than candidate gene studies and linkage studies in the medical genetics world. I should say that they were not considered successful. I think GWAS may be one of the most effective methods that has never had its day in the sun. Basically, early on, the reaction to these early GWAS studies was they didn't find anything. The GWAS doesn't work. And that's because the sample sizes were relatively small. Recently, they find lots of things, and the criticisms are we can't interpret any of it because there's too much, and also the technology is outdated because now everything we should be doing sequencing rather than SNP arrays. But in any case, the successes began to build up in medical genetics in the mid-2000s. In the social sciences, there were basically never linkage studies. And the reason is because we don't have these case control phenotypes that are simple Mendelian traits that you can follow through pedigrees. So you can't do that kind of study. So in the social sciences, the earliest work starting in the mid-90s were these candidate gene studies. And the view was, so that they were modeled after the successes in medical genetics. The idea was, well, we know this ApoE association, these BRCA associations. Let's find similar things for risk-taking behavior or novelty seeking. And the view was that they were, I think the view was generally pretty positive because people were finding big associations and finding gene-environment interactions, and it seemed very exciting. So it wasn't until, so even once medical genetics researchers began doing GWAS, in social sciences, we stuck with candidate gene studies, partly because it seemed to be going well, and partly because there just wasn't data that had social science variables of interest and SNP array data. And it wasn't until the 2013 paper on educational attainment by the SSGAC that large-scale GWAS began on social science phenotypes. And I'll talk about the story of that in the next lecture. I should say some of you have asked me about my own background and how I got involved in this, so I'll just, I'll take this opportunity to tell the rest of you. I got interested in genetics in graduate school, at the very beginning of graduate school, which for me was in 2001. It was a time when the Human Genome Project was getting close to completion, and my advisor, David Leibson, was excited about the prospect of genetics ultimately mattering in economics and in the social sciences. And so I started working with him and Ed Glazer, and basically we couldn't make any progress at all early on because there was no data. At this time of the early 2000s, the data sets that had genetic, it was so expensive to measure the genetic data that nobody wanted to share the data. Basically, anyone who had measured it in their sample of a few hundred people, they were just going to study it themselves. Finally, in 2004, we got some money from NIA to do a candidate gene study in collaboration with the Icelandic Heart Association. And so we constructed this panel of candidate genes that we were excited to study. It was like, at that time, the technology was up to like 300 candidate genes you could measure at once. And we, I took some trips to Iceland and coded up the data to they had measured, even though it was a heart cardiovascular health study, they had some variables that would be interesting for social scientists. So I spent time coding them up. And then we basically ran a regression of every outcome. It was like 10 different outcomes on every, each of these, 300 genetic variants. And we were very excited to find something that seemed associated and it seemed to make sense biologically. And then, by that time, so it was started in 2004, by the time we had done the study and done the analysis, it was 2008 or so. So GWAS era had started, and in medical genetics, one of my collaborators was Sean Purcell, who was a statistical geneticist, and he said, given what we know now in medical genetics, we can't publish this yet. We have to replicate it first. And so we went out and got 3 other data sets, including another one from that the Icelandic Heart Association collected, and the association did not replicate in any of those 3 data sets. So, all this time, and it was a total failure of a study. So then the next thing that my collaborators and I wanted to understand was how widespread of a problem this was. And so Chris Chabris, who's a psychologist and a close collaborator, led a project to look for all of the published candidate gene associations with cognitive ability and see how many of them we could replicate in a very large data set. So this is joint work, with David Cesarini, who's here, Jonathan Beauchamp, who's going to be speaking next week, David Leifson, my advisor from grad school, and a number of other people. And so what we did was looked at this review paper that had just been published on all these candidate gene studies, and combined. We had access to 3 data sets that collectively had 10,000 people in them. That was very large at the time because most of these candidate gene studies were done in samples of 100 people or 200 people. Basically, they were laboratory studies where people would be administered cognitive tests, and then they would do the candidate gene study in that sample. So with 10,000 people, we had much more power than the original studies, and all of these associations that were reported in the review paper had relatively large effects, so large enough effects that 10,000 people should have given us, excellent power to detect them. So we were able to look at 12 of these candidates in our data. And what I'm showing here on the x-axis is our measures of these variants, and the y-axis, the estimated effects in terms of IQ points. So for reference, the standard deviation of IQ is 15 points. And then there's 95% confidence intervals shown here. And what you can see is that in all cases, the 95% confidence intervals include 0, which is to say that none of these candidate genes replicated in our data. And in fact, the 95% confidence intervals exclude effects larger than 1 IQ point, which is very small, much smaller than the effect sizes of the original papers. So, this paper, among other things that we saw in the literature, made us worry that this problem might be quite widespread. In 2012, when we published this paper, that was not the general view in the social science community, but there were some observers who were understanding the problems, and in I think a far-sighted move, one of the leading field journals, Behavior Genetics, came out with a new editorial policy in 2012 so it was actually, this was partly in reaction to a convincing set of analyses that were done by Matt Keller in a paper, Duncan and Keller, in 2011, which was a review of the psychiatric genetics literature, that also came to the conclusion that many of the findings in that literature from the candidate gene studies were not replicating well. So this editorial policy made it harder to publish candidate gene studies in the journal Behavior Genetics. You had to have a replication along with the initial result, and you had to do calculations showing that you had sufficient statistical power. That new policy was accompanied by some introductory remarks that I've put the beginning of it here, and I'll just read it because it's pretty remarkable to have such strong language in an academic journal. The editor here is John Hewitt. He wrote, "The literature on candidate gene associations is full of reports that have not stood up to rigorous replication. This is the case both for straightforward main effects and for candidate gene-by-environment interactions. As a result, the psychiatric and behavior genetics literature has become confusing, and it now seems likely that many of the published findings of the last decade are wrong or misleading and have not contributed to real advances in knowledge. The reasons for this are complex, but include the likelihood that effect sizes of individual polymorphisms are small, that studies have therefore been underpowered, and that multiple hypotheses and methods of analysis have been explored. These conditions will result in an unacceptably high proportion of false findings." Okay, so we're going to unpack some of these reasons a bit. I think that the fundamental problem, as highlighted in the Hewitt quote, is that the candidate gene studies, at least for the social science phenotypes, were based on a false premise. They were premised on the idea that there were going to be a few genes of large effects, large enough to be detected in the kinds of sample sizes that were being used, and that we had hypotheses, enough knowledge to formulate hypotheses that would likely help us, identify which of these polymorphisms were likely to be the ones to have those large effects. It seemed plausible at the time that there would be these large effects because of these successful examples from medical genetics, like the ApoE association and the BRCA association. We now know that those are exceptions, not the rule. Those are the things that happen to have been found in the medical genetic studies that had small samples. And what we now know is that most genetic variants have much smaller effects, and particularly when it comes to these behavioral or social science phenotypes, the effects are really small. We also realize now in retrospect that the hypotheses were not so good in the social sciences for the most part. They would be things like, well, we know that dopamine is involved in decision-making in the brain, and so we think it's going to be related to risk-taking behavior. And so we're going to look at a dopamine gene. But the problem is that there's actually lots of most genes are doing something in the brain, and ex post, you could come up, for almost any association you observed, you could come up with a plausible story for why it seems to make sense biologically. So it's just much less direct than those cases where things were successful in medical genetics, like the ApoE Alzheimer's link. Okay, so how should we think about these three problems in gene discovery in relation to the traditional candidate gene studies? Well, from multiple hypothesis testing, there were sometimes corrections for the number of variants that were studied in a particular paper. You do a Bonferroni for the 5 candidates that you look for. But that wasn't always the case. It was often uncorrected for. And there was the problem came up in the form of publication bias, because typically in these data sets, you would measure as many candidate genes as you could, and then you might look at, do a study of particular candidate genes. The ones that weren't associated, you wouldn't write a paper about those. You'd write a paper about the ones that did show up as associated. That would end up in the literature, but you wouldn't have a correction for the fact that there were these other tests done in unpublished studies. We also know now, subsequent to this kind of replication crisis in social science genetics, there's been a broader recognition of replication crisis in the sciences in general, and multiple hypothesis testing is a leading contributor to broader problems in the sciences. So that's not specific to this literature. That's a general problem. Second, issue, population stratification, well, in the candidate gene studies, we were only measuring a few variants. You can't calculate principal components and control for them. That was an invention that came along in the GWAS era when you have genome-wide data. And you also typically don't have the kind of family data you would need to be able to have family fixed effects. So typically what you would do is restrict these studies to an ethnically homogeneous population, it would be called, like, white people. Or you might control for a self-reported ethnicity. And the problem is that population stratification can be more subtle than that. We know, for example, the principal components can pick up variation within the country of Iceland or within regions within many different countries. And there can be cultural differences that lead to differences in the phenotype within countries. So we think that that's unlikely to be sufficient in many cases. Okay, the third problem is low power, and that's such an important one that I'm going to go into more detail on it. So let's do a power analysis calculation. Suppose that we're testing a candidate gene with a significance threshold of 0.05. Suppose that the true strength of the association is an R² of 0.02% of the variance. That number comes from the largest effect sizes that we're seeing for associations with educational attainment, and it turns out with other behavioral phenotypes we've looked at, this is the right order of magnitude for these kinds of associations. And suppose we have a sample size of 3,000, which is actually larger than the typical sample sizes used in the traditional candidate gene studies. It's on the very high end. So what kind of statistical power do you think you have for this kind of test? Well, it turns out 12% is the answer. You could also ask the question, what sample size would you need for 80% power? And the answer is, which is a typical kind of normal threshold that you want for power, the answer is you need almost 40,000 people. So you need an order of magnitude larger study than you're running in order to meet normal standards for adequate statistical power. Okay, so we're going to talk more about the problems now of what happens when you run studies with low statistical power. But first, I want to address some common mistakes, mistaken intuitions that people have about power. So remember what power is. It's the probability that you find a significant result under the alternative hypothesis, so the hypothesis that there really is an effect, and it's calculated using the truth, the true effect size beta. So when you estimate power, you need to know what is, you don't actually know the truth when you estimate power, but you need to have a good guess about what the effect size is going to be. And it needs to be an ex ante. It needs to be before you looked at the data. And one common mistake is to do power calculations based on what you actually found. That's called an ex post power calculation, and it's very problematic. The reason is that if you found something that's a false positive, something that's statistically significant by chance, it's likely that you've overestimated the effect size. And then if you use that for the power calculation, it's going to look like your power was high. So it's going to look like you've had a trustworthy result. So it's just not very helpful in terms of seeing what your power actually was. Okay, so it can be hard to actually get a good estimate of what effect size should be when you haven't done the study yet. But there's a nice paper I'm going to reference in the next couple of slides by Gelman and Carlin that has some discussion of how you might go about doing it, drawing on existing literature and related phenomena to what you're studying. Yeah?
Can you please repeat the point briefly? Like this exact point.
Repeat this point.
Yeah.
So if you use the ex post estimated effect size, and it turns out, and suppose it was a false positive. So then that means it was statistically significant by chance, which means you've overestimated the effect size. Well, if you've overestimated the effect size, you're going to have a large beta hat, And if you calculate power based on that being the truth, it's going to look like you had plenty of power because there was a large effect. So, your sample size was adequate to estimate that effect. So you're going to think you had high power when, in fact, you didn't. So you can't really use what you've observed. It has to be ex-ante information. Okay, so some misconceptions about power. One thing that you often hear people say is, power matters before I do the study. It matters for writing grant proposals so I can convince them to give me the money to do the study. But once I found an effect, Like, it doesn't matter what my power was. So that's wrong. We're gonna go through why it's the case that if your power was low, then you should be skeptical that when you think you found an effect. Another misconception, which is kind of an exaggerated version of the first one, is that it's like if you find an effect despite having low power, it's like you've overcome adversity. It's like your result is even more convincing if you manage to find it despite having had low power. So that's completely backwards. Okay, so one way to think about low power is that it's equivalent to saying that the true standard error of your estimate is large relative to the truth. So here's the way to think about that. Here's zero effect, and here's the truth, beta. So I'm claiming low power means that your standard error is large. So here, this is like a 95% confidence interval around the estimate. I've drawn it around the truth, but in fact, there's going to be some estimate, and you're going to get some confidence interval around it that's related to the standard error. Now, why is this low power? Well, here's a proof by contradiction. Suppose that contrary to this, the standard error were small. So let's say it's like this. Confidence interval is now tight. So that means we know that in expectation your estimate is going to be centered around the truth. So if you have a small true standard error, your estimate's going to be somewhere around here. Your confidence interval is going to be pretty tightly around the true beta. But in that case, the confidence interval is going to exclude 0. And so you're going to find a statistically significant result with very high probability, which means you have high power. So having low power means that this interval is wide. It's pretty likely that which means that with the estimate centered around the truth, it's pretty likely that this interval is going to include 0. Okay, so that's the low power is equivalent to saying you have a wide confidence interval, which is, or at least that the true confidence interval is wide, which means that your estimate is uninformative. Okay, so the reason why that intuition, that way of thinking about low power is helpful is because I think you can immediately see why you can't conclude much then from results with low power. You can't conclude much from failing to reject the null. That's kind of from the definition of what power is. We know that if your power is low, then who knows? If you didn't find an effect, maybe it was just because your power was low. But it also means you can't conclude much from rejecting the null hypothesis because your estimate is just quite uninformative. And this is the thing that people often don't learn in statistics classes or don't have the intuition for. Okay, so we're going to now talk about a few manifestations of this fact that low power means your estimate is uninformative. So one manifestation is what Gelman and Carlin refer to as sign errors and magnitude errors. So a sign error, or what they call a type S error, is when you find a statistically significant result, it's the probability that you've estimated the wrong sign. A magnitude error, or what they call an expected type M error, also they call it the exaggeration ratio, is the ratio of the expected value of the magnitude of your estimate to the expected value of the magnitude of the truth. So it's easiest to think about this if you forget about the expected value and you forget about the absolute values, and just think about this as basically, if these are both positive numbers, it's, if your estimate is like, twice as large or 3 times as large as the true value. And then the fact that it's an expectation is like it's ex ante, that if you find a significant result, it's likely to have been the case that you estimated an effect that's too large. And they show that when power is low, both of these types of error probabilities get large. This exaggeration ratio is actually closely related to the winner's curse, which Patrick is going to go into more detail about tomorrow. So I'm not going to go into the details of the calculations, but here's some figures from their paper that show the type S error rate and the exaggeration ratio as a function of power for a simple test with a normally distributed variable in a univariate test. So with the sign error, you see if power is bigger than 20 or 30%, the type S error rate is actually quite low. But once you're dealing with power below 20%, like in our example of the candidate gene study with a power of 12%, then the type S error rate can start to get pretty high, and it can be pretty likely that you just have the wrong sign on your estimate. The exaggeration ratio, here this light gray line is at 1, so that would be no exaggeration, and it asymptotes with perfect power to an exaggeration ratio of 1. But for any power less than 1, there's going to be some exaggeration in expectation. That's the winner's curse. Even with power of 0.4, the exaggerate already with a power of 0.4, the exaggeration ratio can get pretty big, like 2. So in expectation, if your power is 40%, your estimate is twice as big in expectation as the truth, conditional on statistical significance. And if your power gets below 0.2, these can get very substantial exaggeration ratios. So this is part of the explanation for why these traditional candidate gene studies were estimating large effects. They had low power. Of course, multiple hypothesis testing is going to make all of this much, much worse. In fact, even with what looks, even what would be higher power in a single particular test, you can make lots of mistakes when you focus on the statistically significant one of many tests. Okay, second manifestation I want to talk about is the risk. Yeah.
So for these figures, do these correspond just to a p-value of 0.05, or does it look the same no matter what? Like if you've done Bonferroni, so you just made this point about multiple hypothesis testing.
Yeah.
Was that about multiple hypothesis testing accounting for it with like a Bonferroni correction, or?
No, it was unaccounted for multiple hypothesis testing.
Okay. So if you do account for multiple hypothesis testing, the shapes of these curves.
Yeah, I think I believe the answer is that if this is conditioning on the this is the true type 1 error rate is equal to 0.05. So if you have multiple hypothesis testing, but you do a correction such that you get the error rate back to 0.05, then this would be the picture that you would expect. I believe, but we can look at the paper. Okay, so another way. Another manifestation of the low power problem is that you get a high risk of a false positive. Okay, so how do we think about what a false positive is? Well, remember, power is the probability of significance given the alternative hypothesis. What we want to know to evaluate whether it's a false positive is the probability that the alternative hypothesis is true, that there really is an effect, conditional on having found a statistically significant result. So that is, the question is the hit a true finding? But this requires us to switch the order of conditioning in this probability. And the way we do that is with Bayes' rule. So I know this was on the problem set, so this is review. But as a reminder, for two events, the definition of conditional probability is this. And then we can write out the denominator as the sum of these two composite events, B and A, and plus the probability of B and not A. And then we can write each of these using the definition of conditional probability as the conditional probability of one event given the other times its prior. And then this is the formula we're going to use where the events A and not A are the null hypothesis and the alternative hypothesis, and B is significance. So we're going to calculate the probability of the alternative hypothesis given significance in terms of things that we know, which is the probability of significance given the alternative hypothesis, and some assumptions on the priors. Okay, so we're going to do this calculation assuming that we know what the true effect size is. It's just some fixed number beta, or beta j. We're going to treat the null hypothesis as 0. And I want to highlight this is a highly stylized kind of example, because if we were actually going to do this calculation, and Patrick I think is going to talk about things along these lines, what you would really want to do is specify a distribution of effect sizes for your prior, not fix them for a particular one, but this is going to be useful for the purposes of illustration. Okay, and then we're going to imagine the information we have available is whether our estimate is significant or not significant. That's also, not necessarily the right thing. If you've already run the study, you have more information than whether a result was significant or not. Like the actual estimate, its p-value, and you should be conditioning on those things in calculating the credibility of a result. So the way to think about this is that we're doing a kind of pre-experimental analysis. We're imagining we haven't run the study yet. We're just saying, is it a smart thing to run a study with a certain level of power in terms of what we'll be able to conclude if we find a result that's significant or not? Okay, so using Bayes' rule, we can calculate the probability that there really is an effect given significance, and we can rearrange it to have this form. And then we can do the same calculation for the probability of the null hypothesis given significance, and then divide one by the other. So if we do that, then the denominator turns out to be the same in both cases. So a lot of stuff cancels out. I think this is actually on the problem set. So you get a convenient form, which is that easier to remember formula, which is that the probability of the alternative hypothesis given significance divided by the probability of the null hypothesis given significance. So the ratio of the probabilities of the two hypotheses given significance, we call that the posterior ratio, is equal to the ratio of these two probabilities, probability of significance given the alternative and significance given the null, which we call the rejection ratio, and then the ratio of the priors, the prior ratio. So we can then notice that this ratio, probability of significance given the alternative, is what power is, and this term, probability of significance given the null, is our significance threshold alpha. And so the key quantity then for evaluating the information contained in statistical significance is the ratio of power to the significance threshold. That's what the rejection ratio is, and you can think of it as the strength of evidence that you get from statistical significance, from the rejection of the null. So we had some prior ratio. The amount by which significance should cause us to change our beliefs from our prior ratio to our posterior ratio is this rejection ratio. So here's some graphical intuition for what the rejection ratio is. We have on the x-axis here our estimated effect. here's the 0 point. So under the null hypothesis, which is shown in red, the distribution of the estimates is going to be centered around 0. It's this normal distribution. Under the alternative hypothesis, there really is an effect. There's some truth out here, and what we observe is going to be centered around that truth. So it's going to be shifted to the right. That's the blue normal distribution. Significance means that we're finding an estimate which is in the tail, the right tail. So this is our significance threshold. I'm showing this for a one-sided test, just to keep things simple. So it's in the right tail. So what if conditioning on the fact that we know that the estimate is out here, what's the relative likelihood of the alternative relative to the null? Well, it's the ratio of the area under the blue curve to the area under the red curve, which is the region, the blue area plus the red area divided by the red area. Okay, so that's the rejection ratio. Here's an example. And so this is not a genetics example. This is an example from social psychology experiments. So what I've done here is imagine that we have an experiment with a treatment and control group. And we're going to assume that the effect size is a correlation coefficient between treatment assignment, which is a 0, 1 variable for treatment and control. We're going to assume that correlation between treatment assignment and the outcome we care about is 0.21. So this is a measure of effect size that's often used in social psychology. And a typical number, according to a big meta-analysis of all research, all research in social psychology up to 2003, is that 0.21 was the typical size. It's probably an overestimate due to publication bias. And then what I'm doing is calculating, assuming equal treatment and control group sizes for different sample sizes from 10 to 200, what is power and what is the rejection ratio, assuming that we use a significance threshold of 0.05. So it used to be common in social psychology classes to be taught that a rule of thumb for sample sizes is you should aim for a sample size of 20 people per condition. If you do that, then your power is about 40%. And that power number divided by 0.05 gives you a rejection ratio of 7.7, which means that statistical significance tells you is 7.7 stronger evidence in favor of the alternative hypothesis to the null hypothesis. The normal threshold for adequate power would be 0.8, which corresponds to a sample size of about 65, which is still larger than many experiments that are done. That would give you the rejection ratio of about 16, which corresponds to power of 0.8 and sample size of 0.05. Okay, so now let's do an application to finding genetic variants associated with educational attainment. And I want to go further than just calculating the rejection ratio. I also want to think about what the posterior ratio is going to be, so we can evaluate false positives. So problem that comes up is how to think about what are reasonable priors. So we don't really know. So it's often useful to think about a reasonable range, and I'm going to just go through a very simple kind of calculation. The starting points of the calculation are going to be an estimate of SNP heritability of roughly 20%. So this is our estimate that comes from a method called Gremel, which Alex is going to talk about next week. Yeah.
The estimate then does not come from the same data, right? But
From different data,
Okay.
yes. So and the Gremel estimate is not for an individual SNP. The Gremel estimate is telling us what the variance explained by all the additive genetic factor as a whole on the measured SNPs. So it's not quite the same thing as what comes out of twin studies. It's restricted to what's on the SNP array. And we think that Gremel is telling us that 20% of the variance can be explained by the optimal linear combination of the SNPs measured on the SNP array. The other starting point is I'm going to take as given that there's about 1 million independent loci in European descent populations, and that's roughly the thought to be the case. So even though we measured 2.5 million SNPs on an array, there's, because of the correlation, the LD, there's about 1 million independent units of variation. Okay, so at one extreme, we could assume that every locus associated with educational attainment has the effect size of the largest association that we've observed. That's going to give us an underestimate of the number of loci that are actually associated, because we think most of the loci have a smaller effect. But if we assume that all have the largest effect that we've seen, then since we know that added up they explain 20% of the variance, if each of them explains 0.02% of the variance, then there are 1,000 loci that are associated with education. And so then the probability that a particular locus is associated is 1,000 divided by 1 million, or 0.1%. Yeah?
And just the numbers, like 20% and 1 million, how are they determined?
Well, so the 20% comes from a procedure called Gremel, which doesn't require you to actually estimate the effects of individual SNPs. It's a method of estimating, of inferring the heritability using genome-wide data. And Alex is going to talk about it next week, so I won't go into it here, but that's where that comes from. And the million loci, I think there's different methods of getting at this number, but I think that roughly the way to think about it is you can look at the kind of amount of LD that there is, and and then calculate from that, how many underlying factors there must be in the genome. Like loosely speaking, if you did a principal components analysis on all the SNPs, you would get 1 million of these PCs, and then you would have no more PCs. So that's kind of the amount of independent variation. I don't know if there's James may have a better explanation.
I want to add something that's related to this topic that was discussed this morning. So one way in which they arrive at that number is, in about 2007, 2008, some researchers did simulations where they had simulated GWASs where every SNP was a false positive. And then they said, well, and how many GWASs that we run like that, simulated GWASs, how often do we see SNPs reaching certain p-value thresholds and so on? And it turned out that in that sense, the genome in Europeans, it varies depending on the population, behaves as if it behaves as if you're testing 1 million independent levels. In fact, that's one justification for 5 times 10 to the negative 8, because that is 0.05 divided by 1 million.
Yeah. So I think there were several papers that used different methods to try to estimate this quantity, and I think 1 million was kind of the, roughly, yeah. Okay, so under this assumption, this one extreme of everything has this large effect size, relatively large effect size, then the prior probability of any particular locus is 0.1%. So that's a lower bound on the prior, because most loci surely have smaller effects, meaning that there must be more than 1,000 loci associated with educational attainment. So the other extreme is harder, but and because of this artificial setup of assuming that we have one or the other true effect size, we're assuming that it's that there's only 2 possible effects. So let's assume, for the sake of doing this calculation, and it's somewhat arbitrary, that the effect size is smaller than an order of magnitude smaller than this, are like a null effect. So we're going to say 0.002% is the smallest effect size we care about. So if then we can calculate the other extreme by assuming all the loci have that effect, and we can calculate the number then of associated loci, which would be 20% divided by 0.002%, which would be 10,000 associated loci. which we then divide by 1,000,000, and that tells us 1% is an upper bound on what might be a reasonable prior probability for any particular locus in the genome. Okay, so then we can calculate now, given our significance threshold of 0.05, and assuming now an true effect size of this large 0.02%, for different sample sizes, like I'm picking here 100, which is like the sample size for the kind of laboratory experiment that was often done in these traditional candidate gene studies, 10,000, which might be like the typical social science survey, and 100,000, which would be the size of the first large-scale GWAS in the social sciences that we'll talk about next time. We can calculate power, so 0.05 up to 0.99. And we can then calculate for different prior probabilities. Here's our range, 0.1% to 1%, that we argued was a plausible range. And then I'm assuming some really big numbers that are more like what people in the candidate gene literature were sort of arguing for, not necessarily numerically, but kind of implying by saying these are strong candidates for association. And then what I'm filling in the table here is the posterior probability of a true association given a statistically significant result. So this is calculated using Bayes' rule. So for example, if the sample size is 100, then regardless of our prior, we basically stay at the same prior from a statistically significant result. There's essentially no information from statistical significance. It should make us no more confident that there's a true effect. So why is that? Well, it's because power is only 5%, or slightly higher than 5% here, which means our rejection ratio is basically 1. That is, if the probability of finding a statistically significant result, if the null hypothesis was true, is 5%. That's our significance threshold. The probability of finding a significant result if the alternative was true is slightly higher than 5%, 5% plus epsilon. So it's basically uninformative whether we regarding the alternative versus the null. So that's why it doesn't move our prior at all. Now, there's a few interesting things to notice here. One is, so if we go to 100,000 people, our power is now 99%. And it's still the case, though, that our posterior probabilities are not very high. In this reasonable range of 0.1% to 1%, our posterior probability is at most 17%. So we still think there's an 80% chance it's a false positive. Even if our prior were high at 10%, it's still a 30% chance of a false positive. So I'm going to come back to that observation, but just making a few observations from these calculations. One thing is, it makes it clear that in this world of genetics where we're dealing with complex traits with really small effect sizes, we're going to need large samples to have adequate power. But they also give us another perspective on why we need stringent significance thresholds. And this goes not only for GWAS, but also for candidate gene studies. And that comes from this observation. That's this observation that our posterior probabilities are not that high, even with 100,000 people. The reason for that is that that our power is bounded above by 1. And with a 5% significance threshold, that means that our rejection ratio is bounded above by 1 divided by 0.05, or 20. So at most, statistical significance is going to give us 20 to 1 evidence in favor of a true effect. And if our priors are small, as small as 0.01, which we said was an optimistic prior, that means our pre-experimental odds is bounded above by 0.2. So we're just not going to end up very confident, very persuaded by results that are significant at a 0.05 threshold in a world of small priors. So in a world of small priors, we need stringent significance thresholds for findings to be credible. This is like Carl Sagan saying that extraordinary claims require extraordinary evidence. The claim of any particular SNP is extraordinary, an extraordinary claim. And for genome-wide significance, we have a different story. Now, if we have 5 times 10 to the negative 8th, now our power divided by alpha, our rejection ratio, is bounded above by 1 over that, which is 2 times 10 to the 7th. So now we can actually get much stronger evidence, which is what we're going to need when we have small priors. And if we redo the calculations from the previous table, but now assuming that we're looking at results with genome-wide significance instead of 0.05, what you see is with a sample size of 100,000, you can be quite confident, even with our pessimistic prior, that the result is a true positive. Note, though, that our power is only 16%. So, given our stringent significance threshold and our small effect sizes, we're only going to actually detect 1 out of 8 of the truly associated SNPs with educational attainment. But if we do find them, we can believe them. Now, this actually gives us another important observation, which is, we've talked about low power being a problem, and 80% power is typically what's standard for saying you have adequate power. But in the context of GWAS, 16% power is perfectly adequate, at least if your goal is to have credible findings. Why? Because 0.16 divided by 5 times 10 to the negative 8 is about 3 million. So you're still getting really strong evidence with power of 16% if your significance threshold is 5 times 10 to the negative 8th. So, I think one important bottom line from all of this is that our intuitions are often related to doing studies with the kind of standard effect sizes in the social sciences, which are much larger than what you see in genetics, and often with higher priors. And so our intuitions can be misleading when we try to apply them in this world of gene discovery. And it's especially important then in this world to do power calculations and credibility calculations for what you find, because our intuitions can be off. I mean, I think the replication crisis that's happened more broadly actually suggests that our intuitions might be off even in the world we're used to. So I think this lesson applies more broadly, but certainly true for gene discovery. Yeah.
So going back to the figure that I was asking you about at the beginning, where it's just you had power on the x-axis and the type M on the y. So if we're getting like 16% power in the best case scenario, I don't remember exactly what it looked like, but at 16% power, I think it was like a 5 times inflation or something. It was huge, right?
Yeah, let's go back to that. That's but that was.
So that was why I was asking about, does it matter what the p-value threshold is for the shape of the curve? Because I don't think that in practice we see things that large, but
yeah, that's a good point. So we can look into it. This is going to come up in Patrick's lecture on the winner's curse, because it's going to be dealing exactly with the problem of exaggeration of the effect sizes and how you would, given what you actually estimated, what's your best guess about what the effect size really is. Yeah.
Maybe my question is complete nonsense and just reveal that I don't understand what you are talking about. That, given those problems with Bamford corrections and power calculation, when you have an ex post estimation of the effect size, it's not similar the problem that we will have with ex post treatment definition, because like in this case, you don't define what your treatment is in advance, but you like the combination of loci that you are assigning as in your GWAS, that's selected ex post based on your results. So is there like a clear hypothesis that you are multiple testing, or actually like you always trying to do this exercise came up with some combination of places that predict your outcome? I don't know if I'm explaining this
So I'm not sure I'm quite following.
Like in the ex post treatment?
Like in the causal model that you define, you say like, what are your like potential outcomes comparison? Let's say that. What are your counterfactors in this? You say is it additive, but additive with respect to what? So you have like I don't know how many millions of particular like SNPs, but you are testing like combinations of those as your final, association.
Are we? No. So I think we are testing all SNPs separately and all of them. So you're not really testing any combination of the SNPs. You're testing all SNPs.
So this calculation was, for a particular SNP. So it's a regression of just the phenotype on that particular SNP. I think there is a deep connection between, this issue of the exaggeration ratio and the issue that, of deciding in an ex post, like what you're going to focus on in your analysis, like how you're going to define what the treatment is, because our beliefs about unbiasedness are premised on an assumption that you haven't conditioned on anything. And if you're conditioning on there being effects that you found, like conditional on statistical significance or conditional on this way of defining the treatment, you're seeing an effect as opposed to not, then our conclusions that we draw from the unconditional model don't hold. And in fact, you can tend to get exaggeration of the estimates. Yeah.
I actually just want to go back and try to understand the prior ratio calculations you had. Because the 20% estimate that you got was based on that Gremel method you mentioned, which is a multivariate, whatever kind of estimate of the SNP contributions to the phenotype, right?
It's an estimate of the additive genetic, the variance explained by the additive genetic factor, where you restrict to the measured SNPs, roughly speaking.
After the fact, and you're using that in your power analysis itself. You see what I'm saying? That exactly. I just feel that there's a little bit of circularity that I don't completely understand, where you had something like a Gremel estimate, and you're trying to use that Gremel estimate to then find the power for a future study of the effect sizes of the SNPs. You see? I'm just trying to separate out.
The estimate was on a different data set, right?
Yeah, so that's right. So that helps. It's also, the Gremel estimate is being used to calculate a reasonable prior. So it's not being used to calculate power, just to clarify. And although it's true that the set, so I don't think that there's the problem you're highlighting, because the set of SNPs that are measured on the SNP array, which is what the Gremel estimate is telling us about, is exactly the set of SNPs that we're potentially thinking about, at least if we're doing this calculation for a GWAS study. So we have from some other sample some estimate of the sum total of the variance explained by those same set of SNPs that we're now in a different sample going to test SNP by SNP. Yeah.
So it's like parsing how you distribute that overall burden of.
Yeah, that's how I was backing out a prior.
I see.
Yeah.
I mean, there would be a problem if we were like trying to pick the phenotype that we wanted to study, and so we did Gremel on like 45 different phenotypes, and then chose to do the one then do a power calculation based on the one that had the biggest estimated heritability. But that's not. I don't think that was the point of the illustration here.
Yeah. Okay. One more question, and then we should take a break. Yeah.
Just from my conceptual understanding, the calculation that you've shown.
Yeah.
Comparable to kind of a base vector, but then for tailored to power?
So it's like a Bayes factor. For those of you who are familiar with so it's like a Bayes factor, except that a Bayes factor conditions on the results of an experiment. So you might condition on, for example, the p-value or the estimated effect size, whereas what we're conditioning on here is statistical significance. So it's the kind of pre-experimental thing. We don't actually know what the value was. But and actually the Bayari et al. Paper, which I'm a co-author on, which I cited here, where we label this the rejection ratio, that paper actually shows that there is a link between Bayes factors and this and the rejection ratio. So you may yeah. Okay, so let's take a 15-minute break now and resume at 3:35.